As you all know, the comet assay is one of the most popular methods for measuring DNA damage, and DNA damage is an important biomarker for human health. And comet assay offers us a sensitive and a simple approach for investigating the association of DNA damage with various factors such as exposure scenarios, various diseases, and lifestyle factors. In exposure scenarios, we can answer the question of whether or not an exposure is associated with elevated DNA damage. And there are several case control studies which are published and use comet assay for this aim. However, to establish the predictive power of comet assay as a biomarker for the risk of a disease, we need prospective cohort studies. And prospective cohort studies require samples stored for decades. In this slide, I would like to share you with a table, one of the recent publication from Comet Group, and this table is nicely summarizing published case control studies and this and the relationship between DNA damage and chronic diseases. And as the outcome of this table, we can say that comet assay can determine approximately twofold increase in humans who are affected by diseases. However, we cannot say whether the DNA damage is a cause or a result of this disease. And to find out more, we still need to answer the following question. Is it possible to use comet assay as a biomarker to determine increased disease risk earlier in life? And for this, best approach is using biobank samples. Because biobank samples have follow-up regarding diet, lifestyle, and medical history, they offer information like body size and weight measures. They have electronic health records, for example, death and cancer registries, and they can offer several types of biological samples. Although for our aim, the best or most suitable sample would be the blood samples. And most biobank samples are stored as buffer code in straws. Together with this, we have two issues it's raising. First one, all studies with frozen samples have so far been done with isolated PBMCs or whole blood. So we have limited experience with buffer code samples. And the second issue is that comet assay studies have so far been focused on recently frozen samples up to 1 to 2 years. So we have limited experience again with decades old samples. Another important issue, what I would like to remind you here, differences between isolated buffer code samples and PBMCs. So we can use 3 different blood preparation in comet assay, like whole blood, buffer codes, and PBMCs. However, there are clear differences in cell distribution, longevity of these cells, and possibly repair activity of these cells. And this can affect the outcome of the comet assay. So we still need to understand which kind of sample matrix would be ideal for human biomonitoring. And together with this background, we have raised several questions that we needed to find answer in our pilot run. First question was, can we perform the comet assay with buffer code? Second question is, are there any differences regarding DNA damage between blood preparations? And third question, can we successfully use decade-old samples? After answering all these 3 methodological questions, we still need to find out 2 important questions to guess the predictive power of comet assay. And first one, is DNA damage stable over time for a particular individual? And second important question, can we identify high-risk groups or high-risk individuals within a group by using comet assay? So before going on with the results from the biobank study, I would like to we have done also some other experiments in order to find out the effect of storage and transfer conditions. And I would like to share this with you. And why we have done this? Because biobank samples are very valuable and they are in limited numbers. And we did know some aliquots from frozen samples were already thawed and refreeze. So we wanted to find out effect of this and whether we can use the samples after thawing if we transfer between the labs. So we use for this frozen blood samples either directly after thawing or 2 hours on ice after thawing, 36 hours at 4 degrees after thawing, or we thaw the samples, freeze them, and rethaw them and using comet assay. You see on the left side of the screen the outcome in alkaline comet assay, and we could observe elevated basal DNA damage except directly after thawing all other 3 methods. So this clearly indicates that storage of thawed samples or using rethawed samples is not feasible for our aim here. What we have done, we also collected blood samples from 20 individuals to test the effect of cryopreservation and suitability of different blood preparations. And we run comet assay, alkaline comet assay and FBG comet assay with these 20 blood samples, either fresh or cryopreserved. We use whole blood, isolated buffy coat, and isolated PBMCs. These samples were maximum frozen up to 1 year. So cryopreservation up to 1 year here in our study did not cause any significant increase in DNA damage, neither in alkaline nor in FBG comet assay. We could use fresh and cryopreserved samples successfully. And among all 3 blood preparations, we could also use all of them successfully in both assays. However, we could observe that PBMCs show a slightly higher basal DNA damage in both endpoints compared to buffy coats and whole blood. As next, we have established historical control by using TK6 assays to observe day-to-day variations in electrophoresis and possible influence of cryopreservation. And we have either not treated or treated the cells with potassium bromide. And after that, we divided the cells into small portions and preserved them in -80. For each time, we pick up 1 pair and taught and run together with our experiments. And you can see from this table, this graphic represents from 6 different runs. So as you can see, we have acceptable range of variations due to cryopreservation and day-to-day variations in electrophoresis. So we use also these samples in all our runs with biobank samples. After this, we have received samples from European prospective study into cancer and nutrition with the shorter names EPIC study, which is one of the largest cohort study in the world with more than 500,000 participants. And they have recruited samples across 10 European countries, and they followed them for almost 15 years. By now, the number of cancer cases might develop in EPIC cohort is more than 100,000. Of course, this is making this EPIC cohort very attractive for our aim. And the German Cancer Research Center in Heidelberg is a part of the EPIC study with about 25,000 participants. They had available samples as buffy coat preparations in straws or cryotubes. And this was available at the beginning of recruitment at baseline level and at the follow-ups. And they had also isolated PBMCs, but only from follow-ups. And we have received from them 299 samples and run FBG comet assay with those samples in order to find answer for these 2 questions. One is stability of DNA damage over time, and second, stability of DNA damage within individuals between sample types. We had PBMCs from 99 individuals at 2 time points. First time point, re-invitation time point 1 and re-invitation time point 2. And these 2 time points were 1 year apart. And secondly, we had buffy coats from 50 participants at the beginning of recruitment and at re-invitation time point 1. And these time points were more than 10 years apart. Sample selection was done according to the following criteria. We had 42% long-term smokers. We had equal gender distribution, 37% normal weight, 43% overweight, and 20% obese. In addition, we have added 47 cryopreserved PBMC samples from our own collection with morbidly obese subjects. And together with this, we were able to cover up all the BMI ranges from healthy to morbid obese. So here in this slide, you can see the first result in FBG comet assay. On the left side, you can find the buffy coat samples, and right side, the PBMCs. And buffy coat samples, compared to the baseline and follow-up re-invitation time point 1, they showed increased DNA damage over time. Whereas the PBMC samples at time point 1, re-invitation time point 1 and 2, they didn't show any increase in DNA damage. So actually, PBMC samples here looking promising for using as matrix for human biomonitoring, because they show stable DNA damage over time. But I represented the results in the next slide in a different manner that you can see it better. On the left side, we have again buffy coat samples and x-axis representing the DNA damage levels from the baseline at the beginning of recruitment, and y-axis representing the DNA damage from buffy coat samples at re-invitation time point 1. And according to correlation analysis, there was no correlation or very weak correlation between 2 time points, which is an indication that DNA damage wasn't stable over time in buffy coat samples. But the good news is for PBMCs on the right side, between the reinvitation time point 1 and 2, there was a positive and a good correlation which showed us stability of DNA damage for PBMCs, which is also suggesting that PBMCs might be a good matrix for using in prospective cohort studies. As next, I would like to share you results from our additional 47 samples from morbidly obese subjects. So when we pull the all EPIC cohort together, they have BMI values lower than 35, and our additional 47 samples had BMI values higher than 35. And if we compare in our FPG comet results, we can see elevated basal DNA damage as well as oxidative DNA lesions with a higher BMI ranges. So comet assay results showed here higher DNA damage level for morbidly obese subjects, which can be an indication for us that comet assay can determine higher risk groups. To sum up together, actually cryopreserved samples were suitable for determining DNA damage, and good news is long-term stored biobank samples can be used in comet assay too. And we have observed DNA damage data from PBMCs shows consistency over the time for an individual, which is making PBMCs an ideal matrix for using in such studies. Unfortunately, buffy coat samples didn't show, DNA damage in buffy coat samples didn't show stability over time. But for this, we might need to develop a further understanding what are the reasons or can we improve the protocol. From our morbidly obese subgroup, we could actually show that comet assay can determine higher risk groups. However, we still need a decoding of these samples for understanding differences between sick and healthy or exposed and non-exposed groups among the EPIC cohort. So with this, I am at the end of my talk, and I would like to first of all thank Dr. Kühn and Professor Cox from German Cancer Research Center for supplying the samples and making this study possible, and Dr. Katzke and Solar for performing a statistical analysis. I would like to thank a working group of Helge Stoffer, my colleagues, for helping me pipetting hundreds of mini-gels and scoring the comets. And I would like to thank also organizers for this opportunity to presenting these results, and especially to HCOMET for great networking and fruitful discussions, which brought all of us one step closer in understanding of limitations and possibilities of COMET assay as a human biomonitoring tool. Last but not least, I would like to also thank you for your attention.
There's one question at the moment in the Q&A asking if the difference between the two BMI groups, was it significant?
It was significant, yes. Since mostly statistical analysis done by Heidelberg, I have done this basically by myself by t-test, so that's why I didn't want to put the stars without their conformational statistical analysis, let's say, but it was significant with a basic t-test.
Yeah, you saw that in the blood mononuclear cells, the signal was stable, whereas in your buffy coat it was not. How do you explain that?
Well, it is hard actually. I was quite surprised. I wasn't expecting it, but first, one thing that I observed that the straws and cryopreservation in tubes. So we had two different cryopreservation preparation for buffy coat samples, and PBMCs were more homogeneous in that sense that they were all in cryo tubes and similar numbers. And for buffy coats, we had also highly differing cell density. I was thinking maybe this might be an effect whether you thaw the samples in straws or cryo tubes, and the density of the cells during thawing might be kind of side effect, but we cannot change it because the samples were prepared like this and stored like this. But plus this, of course, we have a different cell population, and it might be that this difference in cell population is contributing in differences in DNA damage, also maybe due to the repair activity of this subgroup of cells, which we don't have in PBMCs. So I think we still need to go on and try to understand why the different matrices are reacting different. I also tried later on with our own collection in 20 samples, and I also couldn't see correlation between buffy coat and PBMCs in that study. That surprised me too.
I was just wondering in the the last slides, when you compared obese versus non-obese, so below and above 35, but in the 99 people, you had also people which were overweight and normal. So there was
Yeah.
any difference between these two groups?
Since they were still coded, I have no idea. I'm very curious about it, and I hope that we can find out whether there is a threshold that after a certain level we see real increased DNA damage. As Roger already shared with us the repair, it might be correlating in that sense that after 35 BMI they have not so good repair and elevated DNA damage. So we will find it out after decoding.